So by now you're probably pretty sick of seeing this, but let's do a little review. So remember the central dogma of molecular biology. DNA gets transcribed to form messenger RNA, which gets translated to form a protein. So for the most part, one gene, one component of our DNA that makes up a gene, will code for one single protein. Well, humans, we have 23 pairs of chromosomes, and this is like a ginormous amount of DNA, and every single one of our cells has every single base, okay, that encodes our entire genome of about 30,000 genes and billions of different bases. Every nucleated cell in our body, and don't forget, in humans, every cell has a nucleus except for your red blood cells, they don't. So every nucleated cell has this copies of 30,000 genes. If your cell made all of these genes, if every cell in your body made all of these genes at all times, this would require a huge amount of energy. You would have to eat like 25 hamburgers at every meal. It's just like insane. So our bodies can't constantly be making every single protein at all times, especially for humans because we have so many different types of cells and every cell is very specialized. Like a muscle cell doesn't need to make the same proteins that a bone cell does, and a neuron doesn't need to make the same kind of proteins that epithelial cells do. So our cells need to have a way to control their gene expression and turn certain genes on when they're needed and off when they're not needed, especially enzymes. When we need to make certain enzymes for certain functions, we have to be able to turn the genes on and off. So a lot of this control is at the transcription level. Should we make the RNA? Should we even transcribe the RNA that's needed to make the protein? Okay, so can we basically turn this process on or off? Now this process, of course, in eukaryotes like humans, we have so many different types of cells and they all vary so much. So it's really complex in eukaryotes. So early studies, and even today, most of our studies on gene expression are done in bacteria and prokaryotes since they can grow so fast and they're so much easier to study. So today I'm going to talk to you about control of gene expression, and we're going to focus on the prokaryotes, bacteria. So recall, in the very basic process of transcription, you have this DNA, the gene, you have a promoter region, and that's where the RNA polymerase is going to bind. RNA polymerase is the enzyme responsible for transcription. The RNA polymerase is going to move through the gene, transcribing it, making a copy of messenger RNA, until it hits the terminator region, the end, where the RNA polymerase will fall off, and then it can go on to start over at another promoter region. Specifically in prokaryotes, in the promoter region, which is pictured here in blue, from here to here, there's two regions. There's the -10 element, which is 10 base pairs upstream the end of the promoter here. And then there's the -35 element. That's specifically where the RNA polymerase is going to bind on the promoter. Another unique thing to bacteria or prokaryotes, they have these things called operons. So operons are a set of genes that get transcribed together. So in an operon, you usually have somewhere between 3 to 5 genes, but each one's different. And all these genes are under control, the same promoter, and then at the end, they all have the same terminator at the end. Again, operons are pretty unique to bacteria. Bacteria are actually like way more efficient than humans, because you would think if you need a bunch of genes that work together, you should transcribe them all at once in one operon. Whereas humans, we're so inefficient, all of our genes are like transcribed separately. It's like so annoying. So bacteria are way cool. So anyway, an example of an operon, the one we're going to talk about today, is the Trp Operon. This operon has all the genes to make the amino acid tryptophan. So remember, tryptophan is an amino acid, one of the building blocks of protein. Hey, remember when you get really tired at Thanksgiving after eating all the turkey? Because turkey is very rich in that tryptophan amino acid. So for bacteria, tryptophan requires 5 different proteins to make the tryptophan molecule. And each of the genes to make these proteins are in a single operon. So you have Trp E, D, C, B, A. They all get transcribed under control of the same promoter. Once they're transcribed, they get translated into these 5 different subunits, which come together to make the full tryptophan protein. So in terms of regulation of transcription, genes usually fit into one of these 3 categories. It's usually not all or nothing, but we just generally use these 3 categories. So first of all, a gene could be a constitutive gene. You could think of a gene that's constitutive. It's kind of always on. It's a gene that the organism always needs to be expressing because that protein is constantly needed. It doesn't necessarily mean that the bacteria is making a ton of this protein. It just means it always just kind of needs it for something. An inducible gene, you could think of this as a gene that's usually turned off. So under a normal circumstance, it's in the off position, but it can be turned on when needed. And when I say when needed, I mean under certain circumstances, which I'll tell you about. Finally, genes could be repressible. A repressible gene is usually in the on position. So the bacteria is usually expressing this gene in order to make the protein. However, when it's not needed anymore, it'll be turned off when necessary. So like I said, in real life, it's not usually like on or off like a light switch, but just to make it easier, let's think about it like constitutive genes, always on, you left the light on all the time. Inducible, usually off, but will turn on only when needed under certain circumstances. And repressible, usually the light's turned on, but under certain circumstances, we'll turn it off. Now, for these inducible and repressible genes, we need some sort of something to regulate them and turn them on and off when we need to. And those things that will turn those genes on and off are called regulatory proteins. So this is kind of like the hand that turns the switch on and off. When a protein needs to inhibit the transcription of a gene, this is under negative control. So we need to turn off the light, turn off the gene. It's negative control. The protein that's going to turn it off is called a repressor. The opposite of that, a protein that needs to turn on the gene or promote its transcription, this is positive control. And the protein that will turn on the gene is called an activator. So first, let's talk about negative control, or basically when the gene is in the off transcription position. Now first, let's talk about negative control of an inducible gene. Remember, an inducible gene is normally off. So in a normal state, it's going to be turned off. So under normal conditions, an inducible gene is in that negative control state. It's normally off. Here's how it's off. Now here's the promoter. This is where the RNA polymerase would bind. Right next to the promoter, see this little gold thing? This is a region called the operator. The operator binds to these things called repressors. So write that down. Operators bind repressors. Remember, repressors are going to turn transcription off. So an inducible gene, the repressor under normal circumstances is bound to the operator. Now see how this repressor protein is like this big chunky thing? So even if RNA polymerase, here comes RNA polymerase, it looks like Pac-Man. So even if RNA polymerase comes in and tries to transcribe this gene, this giant operator is going to kind of like boink it off. So the RNA polymerase cannot move through the gene and it cannot transcribe. So under normal circumstances, an inducible gene has its repressor protein bound to the operator. If the repressor protein wants to come off the operator, it needs something called an inducer. An inducer binds the repressor and it pulls the repressor away from the operator. So if the inducer binds the repressor, pulls the repressor away from the operator, now happy little Pac-Man RNA polymerase can come in and transcribe the gene. So that's an inducible gene, normally off. Normally the repressor is bound to the operator, but if it wants to get turned on, an inducer has to bind, pull it away, and then the RNA polymerase can transcribe. Now let's talk about negative control of a repressible gene. It's basically the opposite. Remember, a repressible gene is normally on, normally in the on transcription mode. So in normal conditions, for a repressible gene, the repressor protein is not bound to the operator. It's kind of just floating around. It's not bound. So the RNA polymerase is normally free to transcribe. However, if there's something in the environment called a co-repressor, the co-repressor will bind into this little nook of the repressor. And if the co-repressor binds the repressor, then the repressor protein will bind to the operator. And you have what you had up here. Now the repressor is blocking RNA polymerase, so RNA polymerase cannot do transcription and the gene gets turned off. So that's negative control. Now let's talk about positive control, or turning on expression of a gene. So first, positive control of an inducible gene. This gene, again, is normally off. Now when we're talking about positive control, we don't use operators, we use something else. So remember, here in the green, this is the promoter region where the RNA polymerase will bind. Upstream the promoter, there's something called the activator. The activator is this little blue line. In order for the activator to work, the activator protein has to bind to the activator site. So in an inducible gene that's normally off, the activator is usually not bound. So the activator protein is just floating around in the cell. It's not bound to the activator site. However, if there's something comes along called an inducer, remember an inducer protein turns on gene expression. So if the inducer is present, it will bind to this little nook in the activator protein. And now the activator protein with the inducer attached to it will bind to the activator binding site. This will basically pull RNA polymerase in to this toward that site, and the RNA polymerase will transcribe the genes. Now the opposite, let's talk about positive control of a repressible gene. Remember, this gene is normally on. Transcription is normally on. So in this case, the activator protein is normally bound to the activator binding site. So the RNA polymerase can come in and transcribe the gene. But if something in the environment, this time it's not an activator protein, it's something called an inhibitor. If the inhibitor binds to the activator protein, the inhibitor will basically pull the activator protein away, and now RNA polymerase doesn't come in and you get no transcription. So these are kind of, again, opposites of each other, showing positive control, or basically turning on transcription. Now it's always good to get an example. So let's talk about an example of negative control. Remember I told you about that Trp Operon, that operon to allow E. coli to make tryptophan. Here's what that operon looks like once again. You have those 5 genes that all code for subunits to make up tryptophan. Ahead of the genes, you have the promoter region. And remember I told you there's that operator that's inside the promoter region. Now, if E. coli hasn't eaten in a while, it's starving, it needs to make its own tryptophan because it's not getting any tryptophan from its food. So if tryptophan is low in the environment, there's not a lot of tryptophan going around, then the trp repressor is not going to be bound to the operator. It's going to be just floating around in the cell. The RNA polymerase can bind to the promoter, and transcription of those trp genes can occur. So basically the operon is on, you'll have transcription, you'll make mRNA, and then you'll make tryptophan protein. But now let's say E. coli is in a very nutrient-rich environment, like in your stomach, and it has plenty of tryptophan. It doesn't want to waste energy making its own tryptophan when it can just find it in the environment. So when tryptophan is high in the environment, what happens is there's tryptophan. Tryptophan itself is the co-repressor. So the co-repressor is going to bind to the repressor, and this complex, the co-repressor and the repressor, are going to bind to the operator. So now they're bound to the operator, and the RNA polymerase, here it is, if it tried to come in and do transcription, this would kick it out. It's basically blocking the RNA polymerase from transcribing the trp genes. So now the operon is off. So now's a good time to watch this YouTube video that shows you in action how this Trp Operon works, in case you're a visual learner.